So, hello all. This is perhaps week 4 for all of us, meeting again till week 12. So, again, the introduction part. I'm Jagriti. I'm doing my PhD at IIT Gandhinagar. I'm in my second year and I work with Professor Kala in the Muse Lab. So, my area of interest is basically into tissue characterization using quantitative ultrasound. So, I work with the signal processing techniques, make some phantoms, that is hydrogels which mimic our own tissue so that we can tune their properties, see how a normal tissue is different from an abnormal one. So this is how the stuff that I'm doing. But apart from that, I'm also a start early PhD, so I haven't done my MTech. So I directly joined after BTech. So for that I have to do 64 credits of coursework. So I'm still completing that. Haven't started my research since I was a JRF, so I had some knowledge into this. So last week we didn't cover the RLS algorithm. I plan to cover it, but towards the end of the class, if any one of you present over here has a doubt on it, you can ask me, or we'll cover this first and then that. I saw like last time. I can say that I didn't look up at the lecture of that. So I saw that there are not much of that. It's just some formulas are there and you just need to input it and see how the variation is changing, just inversion of matrices and all. So I don't think so something like that is asked directly, but depending upon the matrix size and all, usually people don't go about 5 cross 5 if it's something that you need to calculate by hand, that's analytically. But yes, so if it's given to us to find an inverse and that to be a matrix of around say 4096 plus 128, then it would be something that okay, you'll solve it using some numerical methods, or you have some experimental knowledge or some empirical constants or so in that state you might have some variations of it, and then you'll actually be able to use a computational power in order to solve it. So you don't need to worry for that much. Just knowing the simple fundamental would also do in that case for ABCD lemma as well as the RLS algorithm. So it's just a modified approach of the least mean squares one, that is LMS one, and also it also goes back to the Wiener filter with some slight modifications in it. So I just like to know the head count of the people who have seen the lecture content for this week. That's on event detection for ECG and EEG. If you guys have seen that, then it would be very fastly that we can go through all the content. If you haven't, then I'll explain. It's like this time I didn't get enough time to make some slides, but I have my notes with me. So we'll just go through them. I have mentioned each and every point that was there in it. So didn't miss a point, hopefully. So you guys can just tell me that it's like did you go through the slides, or it's like the lectures videos or not, or do you have any idea about it? Because most of the content for this sessions for these weeks, that is around 19 to 24 from lecture 19 to 24, they were from it's like had a basic information about the week one, so about the physiological signal, how it is, then how do we take the average of it, what all signal characteristics we need to see, and all like that. So you can just tell me, because since we are just 5 of us are here today, so it would be nice if we talk with each other, and so that the session would also be that interactive. Mm. Anyone?
Yes, ma'am.
You have gone through the slides or videos?
Yes, ma'am. Yes.
Oh, okay. Yash. Okay, I guess he's not there. Sri Naresh.
Yes, ma'am, I'm there.
Okay, so you went through the slides?
Yes, ma'am, I went through the slides.
Okay. You have any doubts?
Not related to slides, but from the assignment part.
Okay, that we'll see it later then. Okay, Sri Naresh, you went to the slides or the lecture content?
Yes, ma'am.
Okay, Shravani?
I have doubts in assignments.
Not full,
Okay.
ma'am, but I've seen few videos
Okay.
and assignment.
Okay. And Ayush,
Yes, ma'am.
you have went through the lecture content for this week, week 4?
Yes, ma'am.
Okay. So we can just open up the assignment for these weeks and see how is it. It's like, is it theoretical or you need to do some solving over there?
Mostly theoretical, 90% theoretical, 10% numerical.
Okay. Let's
As
see.
there
Ma'am,
are
it's
10
based on the
questions and
event
one question
detection.
is only numerical.
Okay. Okay. So, okay, wait, I'll share my screen. Is my screen visible?
Yes, ma'am.
Yes, ma'am.
Okay, I guess these you would be able to solve. Then this is there. Then this is also there. This is also there. Diacritic notch is also there. This is the last one for the notch. This is the frequency for EG. Same. Okay, this one. This one you guys had an issue?
No, ma'am.
You were able to solve it, right?
Yes, ma'am.
Yes, ma'am.
Okay, so which one you all have any problem with?
Ma'am, can you explain the third one?
Yeah, so it's within the algorithms that there in the slide. So what we'll do will, as we do it normally, that we'll go through the lecture content, and from there only you'll get to see that, okay, how it's varying. Won't take time. It would be around 1 hour only today because the content isn't that much. Okay, I'll share. Okay. So we all are looking here for event detection. So as we have been going through, it's like you can just tell me what's a basic example of event detection if it's for in an ECG. So if we want to find the heart rate, what do we look there? The distance in between or which interval? So you all remember what ECG was, right?
Yes, ma'am.
So in ECG, what do we look for if we want to quantify the heart rate?
R, R,
RR interval, yeah. So all this event detection is just for an phenomena, and that phenomena that we will be looking in it, so which has an actual meaning to it, so that we can see what insight do we get from that window. So similarly, in the human body, we have those physiological activities which are going on, but we here have to, it's like we want to notice these activities in non-invasive manner. So we also have some things and measures where we do invasive measurements. For example, in EEG or EMG, we have needle electrodes. We poke them in into the particular muscle of interest or nerve of interest, and then we find the correct measurements for it. But here, for non-invasiveness, it will also help us for doing the measurement again and again in a way that it's not that hectic for us also to analyze, as well as we need to see the comfort of the patient who is going through the procedure, or else patients or the participants who are going through won't also be that willing to do this particular thing. So this again helps. So we'll do this all activities or measure this using the biomedical signals. So it's very similar that we need to have a source, and for each source there will be an event, that's an action that's going through. So if it's like in psychology, there's a very popular, it's like just came to thought of it, a very popular algorithm that's what people say is that execution sorry huh it's like execution and evaluation model. So what happens is, like, here if you have the system, here you have the user, then here you have the output, here you have the input. This is just a basic model of it. the user gives some articulated input to it so that, okay, it has some information so that it will go and process. The input is then given to the system that, okay, what the system has to do. Now here it would be just system will do something on the input, that is it performs something. The system gives an output in the form of presentation. These presentations are what we are actually looking into, that is our biomedical signals. And then the user, that is the doctor who diagnoses it, will be able to see that, okay, what's the observation from it, how he can interpret the particular signal and can treat the patient forward. So this is how it's like all in each processes goes on. So if there is a clear signal of the physiological activity, that is termed as an event. So we'll just go through this. All are very what do you say, basic and revision for everyone present over here. So first we'll go through the cardiovascular system. I hope everything's visible to you. So first, what I'd like to know from you all is that, it's like, can you just tell me how the conduction goes on? It's like from where the deoxygenated blood enters, from where it leaves, and how the process goes on. Either it's from right or it's from left. How? You can just unmute yourself and speak. Even if everyone unmutes yourself, it's fine with me. Quick, quick, quick. We'll be done. Then you guys will be able to go somewhere. Yeah, just explain the basic conduction if you know the heart conduction system. Or else I have to say some names because we are only 5 over here today. Please volunteer. Who can volunteer? Let's see. Yash, can you? Okay. Shravani, can you?
First, it enters the right part of the heart.
Okay, then. Are you speaking something? It's not audible actually.
First, it enters from right side.
Then. So it enters from right side. So goes to the atrium and the ventricle. Then where it goes?
Next to the left end from to the body parts to
Yeah.
pulmonary.
So. Go on, go on.
Next to the pulmonary vein, mom, to the body parts. Oxygenated blood goes to body parts.
It's.
And
Yeah.
pulmonary artery collects the deoxygenated blood and again cycle repeats.
Yeah. So that's what she said, that the basic, if you all would have catched that. So the main difference in between artery vein and pulmonary artery and pulmonary vein. So one is used for deoxygenated, other is for oxygenated. So don't always confuse that. If it's an artery, it will always carry an oxygenated blood. So that was how the conduction goes on. It will get through the inferior and superior vena cava. Already oxygenated blood will enter into the right atrium, then the right ventricle. Through it, it goes into the lungs. Then after the lungs, the process to run oxygenated blood via these veins and arteries, it will come into this left atrium. Then it went to the left ventricle, and then it goes to the aorta into the systemic circulation to the entire body. So here I have some chotawala questions for you. So here you can see there are those valves which are being represented by A, B, C, D. Just it's like, tell me, if A is tricuspid valve, B is aortic, C is pulmonary, and D is bicuspid. Is this correct, or are the organization needs to be changed? A is something, or B is something, or C is something else, or D is something else. So, okay, let's say that, okay, first, what are valves for? What do valves do? Or what do they prevent?
They prevent leakage.
Backflow of blood.
Yeah, yes. So then here, if you see in this image, what is the first A1? Which kind of valve?
Bicuspid, ma'am.
Yeah, and the other name of bicuspid valve is also mitral valve. Forgot to write it. So A is bicuspid. What is B? What is B? I mistakenly wrote it correctly. So B is aortic, C is pulmonary. What is left? D is
Tricuspid.
Tricuspid.
yeah. Okay. So the lower ventricle, the left one, it is so distribution of the blood into the systemic circulation. So these are the veins that are at play. Sorry, vessels at play. So arteries, veins, and capillaries, how they vary, and it's like this. So from heart, the oxygenated blood arteries they carry out. Soon as a pit is utilized by our entire body, then it gets splitted up. So these are the smallest vessels which make up veins or the arteries, which it's actually differentiated. That's a branched, vascularized or mesh network of vessels, and then it's all they are also called as there are also veins which provide the supply to the heart, then it goes to the lung, and so you all said that okay, artery is the one for the oxygenated supply, veins for the deoxygenated, capillaries in between interconnection for them. So this is how it goes through the same thing again, but in a more structured way, that okay how it varies. And in biology we normally have, not in actual biology, but yeah, so if it's something is coming, okay, so here the analogy is quite like this, that if the blood flow is away from the heart, it's like red, and if it's also oxygenated carrying more amount of hemoglobin, it's represented by red, whereas it goes towards the heart or going towards and it has less amount, so deoxygenated blood, less oxygenated blood, so it's vein that is blue. But again in Doppler, if we do that, so there if they say that if it's coming towards the transducer, the blood flow is towards the transducer, they say that it's represented by red, and if it's away from the transducer, they say that it's represented by blue. There are some this techniques which are different. It all came from the stars and galaxy, how we see like that. Okay, coming back to the point. So the same thing over here, that okay, from the heart it goes to the pulmonary arteries at the deoxygenated one, then into the lungs for the purification via the pulmonary veins into the heart. So how it comes from to the heart, that is from the superior and inferior vena cava, that is the two big channels next to our heart, and then to the aorta it goes to the entire body. Okay. So if there's any disruption into the entire process, the cells would be deprived of oxygen. And if they are deprived of oxygen, we don't have any energy for the metabolic processes. So in that condition, they say that, okay, if there's a condition called as brain dead, what happens there is uh, all the cells in your brain doesn't have the proper oxygen supply. Say you have myocardial ischemia or infarction. There also it is represented by lack of the supply of oxygen to all of your body parts or organs. But there's also a type of condition like this, that where okay your body can be revived but your brain is not reviving, so you are able to move or do something, but here from here you are actually numb, you don't have any information, or the brain is not responding, your central nervous system is not giving any feedback to it. So there are stages or conditions like that also. So we'll see the different signals and stages which are associated with it. But first we'll see what is the signal that is associated. I wrote it wrong. Okay, so what is the signal that's associated with the cardiovascular process? That was the ECG signal. But okay. So you all might remember from the first class that it's like this, the PQRST segment. So we'll just do a quick review that, okay, what is the P wave for, what is the QRS complex for, and what is the T for. Each one of you can just unmute yourself and see that okay which one does which depolarization, which one does which repolarization. That's it. This section is all about that. It can be just a little revision for you also, so we'll just go through quickly. Okay.
Uh,
P wave.
P wave is for depolarization of the atria.
Okay, next, who is there? Ayush. What is the QRS complex for? Okay. Shravani?
Depolarization of ventricles.
Yeah. And then who is there? Sri Naresh. What is the T wave for?
Repolarization of ventricles, ma'am.
Okay. And then for the atriums? So there's this common hypothesis that is going around that people say that when ventricles are repolarizing, the atriums are depolarizing. So, but you think of it that if ventricles are repolarizing, when a time that they are relaxing, but they just depolarize themselves, atriums are suddenly depolarizing. So this can't be the case. But like there has been research on it proving that okay, some amount of leakage is there, some amount of depolarization is there. It occurs in the order of some microseconds to milliseconds, but mostly people have agreed upon the point that okay, this segment is mostly the isoelectric segment, or say a reference segment, where we don't observe it's like a sufficient amount of supply from the atriums so that the ventricles can get fulfilled. Once the P wave occurs, there only the depolarization of atriums start. So it was something like this. Oh, what happened? Wait, huh? It went somewhere. Okay, it came back. Okay, back to its position. So I hope it's visible to all of you. So P wave for atrial depolarization, that starts from the SA node that we called as a natural pacemaker, and then throughout the atria. Then we have this small PR segment in between the the end of the P complex, that is the end of the depolarization, and in between of the ventricular depolarization. So it reflects the time delay that takes place from the atrium to depolarize or repolarize to start its repolarization to the ventricular activation, that is ventricular depolarization. Then QRX complex, we all know that's a ventricular depolarization. So this is the largest complex that we have, where ventricles has most of the muscle, hence a stronger signal over there. Then ST segment is here, the yellow segment. The ST segment, it's an interval in between ventricular depolarization and the ventricular repolarization. That's where the T segment starts. And T wave, as you all said, that's a repolarization of ventricles. So there are certain, it's like the amplitude of the waveform, as well as the time seconds it lasts for. These are being given. So these quantities are something that if you are working in this area, or if you're giving some sort of exams like GATE or so, these questions are kind of sometimes they might ask, sometimes they don't. So it's something that okay, you logically should remember okay, which is the longest wave. Say here you can see that T is the longest wave, then P comes, then QRS comes, then there are these segments. So you can see ST segment is greater than PR segment. So these all type of questions can come. So from there you can just randomize that, okay, not randomize, but guess that okay, what can be the milliseconds or time duration associated with it. Similarly, which waveform has the highest amplitudes at the QRS complex, obviously, but in some conditions you might say that it's inverted, or you might see that this Q, instead of being negative, it can be positive, but it's overlapping with the R complex. So these are associated with some pathologies. So in GATE, they sometimes they show this for tachycardia, bradycardia, fibrillations, or flutters or so. So there you might need to know about all their pathologies and how it occurs, or what form of signal changes into the signal that you get a difference, how you get a difference. So for P wave, it's usually from zero point 0.1 to zero point 0.2 millivolts, and it lasts up to for 80 milliseconds. So that's a very slow waveform because it actually starts from SA node. So it takes some time for the SA node to also activate. Okay. Then the QRX complex is the biggest one, so it's on the order of 1 millivolt, and it lasts up for 80 to 100 milliseconds. So from here you can see that, okay, if the QRS complex doesn't look as that large as that of P, but again, this is not drawn to scale. But the QRS complex is obviously more than that of the P wave, that's around 80 to 100 milliseconds. So then people also is like, okay, then there are more number of segments. We only saw PR segment and ST segment over there. So we also see the ST interval, the QT interval. So the ST segment over here, in a way, it's important because it represents the pathology for myocardial infarction or ischemia, or if it's someone is depressed or so. That again represents to lack of oxygen supply. Where sometimes if your body is not getting enough oxygen, there might be some psychological as well as physiological factors which are involved in the same process. So if not getting enough oxygen would also be associated with like letharginess or some diastrophy in a way. So it happens. So ST segment is like when the flow of blood is constant and the ventricles are contracted, but then the T wave will, as soon as the T wave we reach the beginning of the T wave, they start to to relax. So this is again, so we do not have any event that's occurring through in this particular phase. It's just the constant phase that was happening and some constant phase that is not even yet started. So this is a non-event phenomena. So this is an isoelectric segment in order to see that, okay, we do not have any information in here. So last time, if you remember, we saw something that if we have two complexes of this PQRS, and if we have this ST segment in between, we can always take this point as a reference because we do not have any essential signal underlying in it. So we can always use this as a baseline or reference signal and can find our information by comparing this particular segment to our original or entire segment and can reconstruct the original signal again. Though we won't get that appropriate or accurate level of precision, but yeah, we do get an approximation of it. So we always want to be lying near our signal as max as possible. So it's like that. So this ST segment is like 100 to 120 milliseconds over here. That's it. So apart from representing this PR interval, people also give this PQ interval. That is start of the atrial depolarization to the start of the ventricular depolarization. So it's important in order to recognize the baseline of the ECG, that where your actual signal will start from, where its 00 is being going on. So it's again difficult. And then this T wave is very difficult to interpret. Why? Because this, as we saw initially. So this is also the case with arteries and veins, that not, I guess, with arteries and veins, the one is thicker, other is thinner. Yeah, true. Which carries blood with enough amount of pressure. So arterial is like atriums here, they have the walls very thinner. So they just pass the blood what they get. But ventricles are even responsible to pass it into the lung via the veins that we have or vessels that we have. And then they also via the aorta, it needs to push the back all the amount of blood that's either oxygenated or deoxygenated to the entire body or to a vital organ where the purification will happen. So here, these walls are very thicker. So we need large amount of muscles over there. Hence, it's very vascularized tissue. You all know that we have Purkinje fibers, we have those bundles of his, everything, the vascularized network we have there. So hence, it gives us a very strong signal and it's very easy to detect. But in certain cases, this particular wave would be under dependent, but again, it depends that which particular condition and how you're looking through into. So this is the longest wave, that's around 120 to 160 milliseconds. So why the delay? That why do we have a certain amount of time in between? Say why the PQ and PR interval is that different? It's just because of this AV node, because it gives in certain amount of delay, and this delay just because we can complete that, or not we, but the atriums can complete their depolarization completely, so that they get empty and then give the ventriculars, and then the ventricles will process, and hence the cardiac cycle completes. So it just performs or behaves as a buffer, which will hold it for some time, let it be delayed, let the ventricles be relaxed completely. They have enough amount of space, enough amount of volume to account for it, and loses it. So it's important in a way that, but it's not that special. But the activity over here is also not related to any event. So it's just that to ensure that everything is going on well and a process is smoothly functioning. That's it. Also, it's important to get any idea of the baseline and if any baseline drift occurs. That is, if there is any variations of the PQ or in just in case of any pathology. So how to, and then we go into the event detection. So we have completed the physiological signal, that's an ECG, how it comes, what's the anatomy behind it. So we'll see that how to detect it. So this ECG's events, they have very high magnitude, and you can also detect them through the slope of the signal. Just do the gradient of it, that's d by dt or d by dx in however scale you're measuring it. Mostly it's a time because we are measuring it with respect to axis. And then it also boosts the high frequency signal. But if you all, oh sorry, but if you take a derivative of a signal, it boosts the high frequency. What this high frequency part is, high frequency can also be your signal and your noise. So be robust around, or whenever you use this d by dt, be very careful that okay how you're going to use it, because you don't want that your noise should interfere with your signal. So you all can always use the prior information that is given to you, that it, in order to suppress the slow moving or less amplitude signal or high frequency noise, so that what you always get is the signal of interest but not some unwanted information. Okay, so this is the basic model of how people do this ECG event detection, or how do they see a different amount of activities that are going through ECG. Say if a person's ECG is being taken off, but he is given a task, say a treadmill task, or to say, okay, do all your daily activities, but what we need to do is just monitor your ECG day by day. So there's a monitor that just hangs up here on your chest, and there's this disposable or plastic electrode. So through this sticky electrodes, we it's like just stick them over here, and it will monitor your 24-hour activity, something called as Holter monitor. So people have been using them today. It's like nowadays. So it helps us to monitor the activity even when the diagnosed us, like the person or the doctor is not near you. And there have been technology invented where they are trying to make those Holter monitors, which are this big, they're trying to make them small and small and small using some integrated chips and technology. So okay, back to point. So ECG, initially what to do is like take the signal, do the derivative, but here be aware that using derivative, it will also amplify your noise, as I said previously. Then we give some threshold. Now this threshold can be basis on your maximum peak of the signal, but half of the maximum peak, just so that you don't overestimate your signal. You always want to be within the limit where there is an underestimation and overestimation, so that you get your signal properly. And then you detect the QRS peaks. That's it. So this is the derivation based first method. So from here only, the question that you guys asked there, that you had a problem, will start. It's very basic. A simple flow chart is there. So you just, so this algorithm was given by this authors, that is Palda et al. Or Alstrom and Tomstig et al., Freezer et al. And Tompkins et al. So hence the algorithm is named by their name. It is always nice so that you acknowledge someone or give a credit to someone, so that the people are also aware that okay, their work is being used, and they feel nice, they feel appreciated. So it's a good practice if you all gonna go and join academia. Okay, here. So what do you do here is that you, so there's a one difference in this yellow diagram and in this sea green colored diagram. That is, you have an additional amount, it's like addition of another quantity entity, that's a filter added over here, just because we need to it's like go out of this noise regime. We don't want this noise. So what do? We take a ECG. Initially what we were going to do is what we were doing in this first one was that we were taking the derivative operator. Now it will obviously, it will reduce the noise, but the low frequency noise, because what we are going to use is just a low pass filter, not a high pass filter. So then it will improve the SNR of the noise, would be robust to the low frequency noise. But what to do for the high frequency noise? So hence we use a filter for it, which will suppress the high frequency noise. And then similarly a threshold and the QRS peak detection. So here they suggested to take the 3-point first derivative. So what is this 3-point first derivative? So suppose that you have a point, okay, you have a point, this n, go backwards from it, n minus 1, n minus 2. So here n minus 1 is the center. Now you take a difference between both of them, that's n minus 2 minus n, or n minus 2, like that. Eventually you take a modulus, so the sign doesn't matter over there. So this is how you take the first derivative. You all must have seen the order of derivatives. So what we used to do is like x plus h, it's like limit n tends to infinity, x plus h minus x of h divided by h, so where h is going towards infinity. Similarly, so first derivatives method, but that to be numerical derivatives. So then we take the first order derivative for the same. So what do we do is like x of n minus x of n minus 2. Then we do is like x of n minus 4 minus x of n minus 2. What we get is x of n minus twice of x of n minus 2 plus of x of n minus 4. So then through empirical modifications or doing some experiments, these people got that, okay, if we do this weighting like this, have a weighted combinations of 1.3 and 1.1, we might get a nice signal which is able to suppress the noise towards the extent as well as amplify our the signal of interest that we are looking into. So this is something that if you don't even remember, it's very fine, because these are the empirical numbers and constants. So one always doesn't need to see that, okay, how it's going on through or through what these particular constants came. So then, so we found that, okay, we had y0, we had y1, then they saw that, okay, y2 is derived from this particular equation. So they gave this y2. Now there's this third equation, that is y3, that is the third derivative. So one derivative of y2 of n. So what do we do over here is that we take y2 of n as an input, and then we take an output. So how do we find that output? We take 8 points of the y2 signal and then do the moving average filter. So take 8 all points in a certain window and then average all of them. So average or combine. So this is just done for the purpose of thresholding, so that we'll be able to threshold the signal, so that we get a smooth, nicely varying signal. So that was the very first method where we employed filters in it, and we took the 3-point first derivative. Now the second method is like this. The flowchart is very similar, but the threshold would be varying. How we take the derivative would also be varying. So what they did is like they weighted, but they squared the first derivative operator. So when taking the derivative, they squared the waveform so that they don't have any negative components here. They are not just like exploiting the phase of the signal, but what they are doing is like looking at only the magnitude of the signal. Now everything would be positive, and then they did the moving average filter. Apart from that, they did the peak searching algorithm. Now this peak searching algorithm, if you say basically, say normally if you just have peaks, what do you do is like you have a signal and someone said you that, okay, told you that just find the peaks for me for this particular signal. What will you do? It's like you'll just do max of peaks, or there's this command in MATLAB that they will do find peaks. So it will help you find the peak, the maximum amplitude of the peak, as the amplitude or magnitude of the peak, as well as the index at which the peak is. That's the location of it. So if you do in that way, it's also fine. But if you set some threshold for the same, it's also fine. So there are different algorithms in order to find peaks. It also depends that which one do you want and what's the reason behind it. So those all things are there. So after next, there's this Pan-Tompkins algorithm, which is usually, it's like Pan is another person, Tompkins is another person. So, but it is known by both of them. Usually people say Tompkins algorithm. So this is a real-time operation that's very nice, so that we'll be able to detect the signals and people would be able to just to see that, okay, just acquire the data and just we are able to see what the interpretation of the data is back then. So what it utilizes is the slope, amplitude and width of the QRX complex. So here, there is a lot of changes. So we take ECG signal. Apart from taking just a low pass filter, we take the band pass filter, differentiate it, do the squaring operation, and then the moving average filter and find the QRS peaks. So band pass filter is officially a combination of a low pass filter and a high pass filter. So the response of band pass filter is like this. So just so similar to a Gaussian, but not that ideally as Gaussian. Then derivative, what it does, we know. But squaring operator is just a nonlinear enhancement of the signal amplitude, so that it makes everything as positive, which will emphasize the larger derivative, a larger slope difference. So there you can actually see what's the variation of the slope. And then you have the moving average filter. Now this moving average filter, we all know that what averaging is. It's a type of a low pass filter. So what it does is also a smoothing. So we have multiple filters or multiple peaks. You take 30 of each and you average all of them. So there's this magic number called as 30, n equals to 30. You all know what's the reason behind this? It's something related to stats, but people always say that, okay, if you need to get some significant results or differences in your data, you always use 30 samples, n equals to 30. If you take lesser number of samples, it's also fine. You have statistical techniques to do so, but taking 30 is something that given in research papers or even this grant agencies and all that we give, they like minimum 30 sample size they require in order to show that, okay, this is something validated and will be able to perform the studies ahead. So you know what's the magic number all about? You can just randomly say anything. It's fine. Yes, I told you. But this number just came in and it just told us that okay, how we can see the differences and just a minimum number. So there are this test called as t-test, ANOVA, and all of them. They require certain number of samples, and for in order to reach that significant differences, a certain degree of freedom is also required, so that if you change one point to another point, you have a flexibility of moving around so that you can get the differences. So it just so occurs that if taken n equals to 30 as a minimum sample size you can take in order to get significant results. That's it. There's no particular technical reason behind it. So yeah, 2 minutes. Okay, so next. So this type of filter, what they use is an IIR filter that they have defined over here, just in the case because it uses the poles and zeros and the weights that are given as in the order of 2 raised to n. That is, it varies. So if you say over here, that is 32, so it's not actually 2 raised to 5. It can also be 2 raised to negative 5. So the weights can shift, so they can shift. So in a way, if they shift, delay, or it can be advanced. So here. So there's a low pass filter, and this is the transfer function for the low pass filter given. Don't ask me why or from where, because this is just a definition given and just an example from their own experiments, and they have designed a formulation for it. So it's like 1 by 32 of 1 minus z raised to minus 6. So I hope you all remember z is the transform over here, but not just a basic variable. So then we formulated the difference equation for the same. So saw that okay, how much it's delaying, and then just wrote it in a similar way, so that if you take y of z by x of z and then take the inverse z transform of it, you get this difference equation. So same as I said, that if it's 2 raised to 5, it can be shifted towards the right side. If it's I guess I wrote it ulta. Huh. And then if it's 2 raised to negative 5, it is ulta. Perhaps it's ulta. Huh. Then it's the advance that is shifted towards the left hand side. So again, if you do, it's like if you take advance of something, that is something that you expect would happen before the time that it's being expected to happen. So that's a very relatively a faster operation. So here they propose that the center frequency of this particular algorithm was 11 megahertz and the sampling frequency was 200 megahertz. So at a particular frequency, you get, it's like in a particular cycle, you get 1 by 200 times of sample points or data points there. So now, if your frequency, the frequency that you're using through, or any frequency that you're going through varies, then that particular, like say the noise or the supply noise, something that's high frequency would be eliminated, and hence we are easy and good to go that we do not have any frequency with us. Again, so these people, they saw that okay, at 60 hertz, the attenuation was greater than 35 dB. Here, I would like to know that if you all know what attenuation is, because that will only help you know that, okay, how we are able to suppressing this power frequency.
Attenuation means some loss of signal.
Yeah, so it's basically that, say if you have, so as I work in ultrasound, I'll give you in context with respect to ultrasound. So if you pass, yeah, it can be related to ultrasound. So if you pass some ultrasound signal through, say, some soft tissue, say if it's my hand placed in there, and initially you saw that, okay, what was the signal response if you placed a perfect reflector in it. So you know what's the response of it if you plot it. And then if I place my hand in between, there would be some absorption through my hand. There would be some absorption, some scattering, some attenuation that would be playing a role into. So here, what if we compare the first signal, that's a reference signal, with respect to the signal where my hand was inserted into, we'll see that the amount of signal has been reduced. So there is a loss of the signal. So we no longer represent this loss as negative of something. Just in case that we say that, okay, it's a loss, we just say a positive quantity, that's a magnitude of it. Or similar for decay also, we never say that's something negative times itna decay present in the signal. We always say a magnitude of the signal and then say that, okay, this is the amount of decay that we have. So if you use a 11 hertz frequency of a signal and frequency any greater than that is directly eliminated, you are free to go from the suppress the power line frequency, just in case our frequency is 50 hertz. So do you all remember that what was the basic filter in order to remove a single frequency? See here, if it's 50 hertz, or outside there it's 60 hertz. A filter that removes a single frequency. I hope you are able to understand my handwriting. Sorry. Any one of you? What's the filter type of filter which stores or removes a particular frequency? A single frequency.
Notch filter.
Yeah, that's it. So this question mark is notch filter here. Okay. So since we saw that, okay, we needed a band pass filter over here. So in order to make a band pass filter, these guys, they made a separate low pass filter and a separate high pass filter, and then found the difference equations for the same. So this is how they gave the low pass filter by, founded the difference equation, high pass filter, their difference equation. And then they saw that, okay, this, if the center frequency for it was 5 hertz, and typically the QRS energy, so not typically, but coincidentally, the QRS energy was also in between 5 to 11 hertz. So if we actually use this particular algorithm, we'll be able to get the particular signal very nicely. And actually this is a linear operation, so you do not have any nonlinear enhancements over there also. So again, it's also an integer filter. So in the order of n equals to x, or say n equals to z, you will get all your operations being done. So similarly, the difference operator for it, they made it. And again, it can be, it's like the multiplication, you can change it in with respect to the shift operator, say advance or delay. So what did they contributed then? Okay, so apart from adding a filter or making it a band pass filter, or making it seeing that, okay, it was coincidentally lying between 5 to 11 hertz, and it just helped us with the frequently. Or last time I guess we saw that how we update the weights with respect to some timestamps. Now here what we'll see that with respect to the initial values of the previous signal, how do we estimate the values of the current signal, so that we'll be able to estimate the values of the future signal. So it's something that's updated constantly and simultaneously for the coming on process. So here there are some notations. The signal peak is represented by this SPK1. Then the noise peak is represented by this NPK1. Then for the peak one is the signal peak. And there are two thresholds that they have defined. So for the first one, these are constants, they because like it's all from the empirical. So there's no actual logic behind it. It's just that you can see that how much it varies, and from all of it varies. I just saw, okay, it's 1/8 of something, it is 7/8 of something, but you do not have actually your entire signal to be 8th of something. But just assume that if it's 100 and this 100 is equivalent to 8, it is represented like this. So this is 0.125 of the peak one, this is 0.875 of the spatial peak. Now, something like this won't be asked in your exam. It would be just that you need to know that how the signal peak and noise peak would be varying. Again, for the noise peak, it's similar like this. So then for the threshold, this first estimate of the threshold. So now, as I said earlier, that this thresholds should always around your signal, it's like the maximum peak of the signal, but should not be that directly towards a signal, should be half of it. So what they did over here is that they took the noise but made it so that they are able to eliminate the noise. So made it above the noise floor, less than the signal peak. So took the difference over here, so that here we are eliminating the noise, and then added a 0.25 empirical constant in front of it. Then the threshold 2 was just half of the threshold 1. That's it. So from there, again, if this equation doesn't work and we do not have our values in between these two thresholds, they saw that, okay, we can also use this 0.25 times of peak 1 and 0.7 times of the signal peak 1 using the threshold 2. Now here, the estimates were based on the threshold 1 for this particular case. But now here, the estimates are based on the threshold 2. Okay. So doing this way, what they saw that we'll update it again and again, update it again, again, again. So we are able to get a precise approximation of the signal that we are looking into. Hence, we're increasing the speed and we are getting a, it's like faster estimation. So again, after that, it's like, so this algorithm was called as adaptive thresholding. So the next one is search back technique. Again, similarly, we had, so since we are looking into the RR waveforms and the average and the heart rates of it, because actually what you're going to get is you won't officially differentiate the P wave from the first waveform and the P wave of the second waveform. You can, but there are a lot of signal processing techniques involved in it. You need to know the shape of it, the frequency of it, the duration of it, and many other things like the features of it, the attributes that it's holding. So just taken, so RR is very sharp and a good spike like waveform. So you can actually see the difference in between there very nicely. So what we are looking to is also the peak searching algorithm. So the peak that we have in ECG is the only QRS peak. So here we took two averages, that is RR average 1 and RR average 2. Both are normally equal if it's a normal condition and may vary if it's an abnormal condition. So here, if you say if it's at X, the value is something, delay. So that's a weighted average of all the past input and the present input times 0.125. So for them in the search back technique, what they gave or hypothesized that always these two values of RR average 1 or RR average 2 should be within these two thresholds. That is the lower limit and the higher limit. And this particular limit is not actually that symmetric. So it's like 92% of the second average, 116 times of the second average. So if you see, it's not a symmetric window. If you see 100%, this 92% has an 8% allowance from here, as well as this 116%, it has a 16% allowance from here. So it actually varies, and it's not very clear that why they took like this, but it's just a window. They saw that, or they want that, okay, values to be lying between them. If it's between them, it's a normal condition. If not, it can be noise. It's an abnormal condition, or can be low frequency noise. But if it's not, again, can be abnormal, can be a noise, or can be a high frequency noise. So this is how the people for the search back technique they gave. Again, we wanted to see something that, okay, if something is missed out and if it's not lying within this limit, then they say that, okay, go back to the previous algorithm that was this adaptive thresholding, and you can just see it's the threshold is in between threshold 1 and threshold 2. So with that, they gave this number that is 166% of the RR average 2. So just gave that, okay, just see if it's like this, then we can say that it's a missed limit. So what you do in those cases, you perform the experiment again, you see, okay, if it's constant, repetitive, you perform some other experiments, or say for doing the pathology of heart, you might do, what else you do for heart activities? Like apart from ECG, you might see actually that how your, okay, sorry, ATMs are working, or you might focus on a particular area. You might use the precordial electrodes, but I guess ECG is the conventional method for this. You can do this one, what's it called? Ultra echocardiography. That's using ultrasound. This is using the electrical signals. That's using the sound signals. So both of them have a nice good resolution. So you can see that, okay, what's varying, or if it's not noise, or if it's not the frequency, or if it's not the some artifacts, then okay, it's something abnormal going through. So we need to analyze it again. So you all know that there are different types of sounds we have, and these sounds are due to the valves which open and close. Different types of valves that we saw previously, they make a sound like lub dub, and this lub dub is like, it varies between the depolarization, that is the systolic phase, or the repolarization, that is the diastolic phase. So there are different kinds of sounds. We normally have, I guess, 3 to 4 sounds. These are normally called as murmurs or so. So now this sounds variation occur, but whenever the sounds are occurring and there is the variation from systolic to diastolic, you see something that is called as a dicrotic notch. Okay, it came somewhere else. It was a perfect. Okay, so there's something that occurs over here, this particular lump or mountain like structure here, that is called as the dicrotic notch. When the aortic valve closes, that's the biggest valve that we have, which sends the blood into this entire systemic circulation. So this is how we have different things, and through which you can also enhance the signal, or you can find the features or significant variations in your signal and see that how it's varying. So you might want to go through them again now. So I just included the technical, or what do you say, the theory behind all the concepts. Sir has given some examples, well, nice examples for the same, that how it varies, or using this technique how do we see, using the moving average how do we see. So there are some good pictures and of the signal and how the signal is varying, how smoothing is being done. So you'll be able to relate it then. Okay, so ECG is done. Any doubts? So I hope that question is being it's like you are it's like you'll be able to solve this question. That was question number 3 of this current assignment, that what was the algorithm based method proposed by Balda et al and later improved by Tompkins and Frentzen. So that's for the QRS peak detection.
Yes, ma'am.
Okay. Any one of you, do you have any questions for ECG now? Because we'll be going into EEG. EEG is very small. Will be done. And then some assignment questions of previous year.
Ma'am, I have a doubt in the
Yeah.
Penn-Tompkins algorithm part.
Okay.
Uh,
Yeah, you go on.
what is the role of the differentiator in the in this peak version, means in the ECG signal?
Oh, okay. So, okay, the ECG signal, it's more like this.
Yes,
And
ma'am.
here you can actually see that the slope is varying again and again. It's not like an EEG signal where you have that nice distinction that, okay, sorry, it's not as EEG signal where you don't have this nice distinction that, okay, how a particular wave is varying and you'll be able to get the difference or not. So here what people do is like they directly differentiate or take the slopes of the signal. I think that you are able to understand what I mean by the slope. It's like they see that how the signal is being varying, where it's constant, how the slope is or the gradient is being varying or not. So through this we'll be able to, it's like if there are, it's like some jaggies over here or there or some wide variations, we'll be able to minimize those variations, smooth it out, remove the noise, and highlight the maximum peaks that we have. Of course, doing so will also remove some part of information from here, but the information won't be removed that in case if that, say, your signal level is like this and you're doing some derivative based operation, if this becomes this level, it would be maximum at this level. So you won't actually would be like, okay, they both are overlapping or so you'll be able to differentiate it. So that's why we normally, what do you do is like, if you have something, people directly find the maxima of it or minima of it. How do we do so? We differentiate it. We do first differentiation, we do second differentiation, find the local maxima, local minima, like it. And just to it's like, okay, there we had a problem that, okay, it was able to remove the low frequency noise but not the high frequency ones, so we used a filter for the high frequency ones. Where it's all correct, right?
It means it suppresses the information of P and T wave and enhance the information of QRS wave, like this.
Yeah, yes, because eventually we are trying to detect the peaks over here, so that is the QRS complex. So we want that it to be retained, but the lower frequency wants to be loosened up, or to noise to be loosened up. That's why we are using.
Okay, ma'am, done.
Okay, so then we'll go into EEG. Someone else has a doubt? I wanted to ask, do you all have your exams going on within this period? Because I have my exams.
No, ma'am. Actually, these NPTEL courses are our semester exams.
No. I'm not asking for NPTEL courses. The curriculum that you all are into, say, undergrad or PG or something. Do you
In
have
this
exams
semester,
within?
this is our curriculum.
This is your curriculum.
Yes, ma'am.
Okay. I hope so that you're able to understand whatever I'm trying to do. Okay.
Ma'am, it's very difficult to understand for fourth year BTech students.
This content.
No, ma'am. Actually, the overall that the lectures are being uploaded.
Yeah, I do understand, because it's again a big week. I myself haven't done an NPTEL course ever. It's like I have watched, but I was not able to go till the exam stage. Even if I gave an exam, I was not able to top in it. So I agree.
Ma'am, if we fail in the exam, then we will be detained for the entire year. There will be a SEM back in our college if we fail this exam.
So these NPTEL courses are considered as a part of your curriculum. So you get credits based on these courses.
Yes, ma'am. There is a credit transfer.
Oh, so isn't it that easy that if you take a course that's just floated in your university instead of taking this NPTEL course? It's like just saying. I don't have any doubt.
It's a law by our college.
You both are in the same college?
Yes, ma'am.
Yes, ma'am.
Okay. What about others? You're here to learn apart from the curriculum or just as a part of curriculum? I guess it's fine. Others are quiet. Okay. So we'll go into EEG then. Okay. So we all saw previously that why we use EEG, it stands for electroencephalogram. So there's something for as an output, and there's something as an instrument, and there's something as a technique, that is graph and graphy. So it has this different features, that is alpha, beta, delta, theta, and gamma. And all of these have a frequency range of around 0.1 to 100 hertz, and their amplitude varies in between 2 to 200 microvolts. So it's like they had a particular okay, it also got changed here. Shoot. Okay, wait a second. Systolic, diastolic, dichrotic notch. Okay, lesion depression. Okay. So for this, if we have gamma waves, we always start from the low frequency ones, that is the delta waves. If you see here, so here the delta waves has the frequency range in between 0.1 hertz to 4 hertz. So you see it's very slow activity. So that's always when you're sleeping very nicely and very strong sleep. So deep sleep, and that's non-REM sleep. So this is something that people use for lesion studies. That is okay, how the activity is varying in this particular region or that region. Then we have theta waves, that is for light sleep, deep meditation, or some recall, or you're finding something creative, something that you're doing. But they also use it for depression studies or finding a metabolic disorder. So these are the two basic pathologies that people use the delta waves and theta waves for. But then after these particular activities, it's like when the person is not conscious, there are many things to monitor in a particular person. But when he's not conscious. So when it's conscious, it's something that it's like, even if you tell someone that okay, don't act something, don't think something, there's something always going on. So your human mind is always manipulating some or the other thing one after a particular point or not. So you're not actually able to see or make sense of the thing. So that's why most of the people prefer doing these activities. It's like when the delta or theta waves are most prominent and you're able to see, or the gamma waves. So these gamma waves are something where your motor functions are very active, there's a high stage of mentalness. Also you'll be able to see all the neurodegenerative or functional disorders within this particular range of frequency. Then we have this alpha waves, that is for the relaxed light meditation, creative super learning conscious state. Then the beta waves, that is for normal walking stage, all your 5 physical senses are working on, that is touch, sense, hear, vision, all of those taste. So concentration, focus, and everything's integrated. So this is how EEG frequencies were. And now there are different complexes in EEG signals. So I also got to know about them recently only, that I didn't know that there are these different EEG complexes in EEG signal. Okay, we'll see. So all of them only depends upon how the particular signal is, how the shape of the feature is. That's it. So the shape is something that through which they get their own name. So if it's a K complex, if you see here, it's not actually looking like a K, but this K complex is something that occurs when you give a response, if you give a stimuli to a person who's sleeping. So before a sleep bundle or after this sleep spindle, it refers to a stage where a person is actually within a state of mind where he's active and not active. So a state of consciousness and unconsciousness. So that's a K complex. It's a very slow wave, but a very sharp one. It has 14 hertz frequency, and it lies like the amplitude of it is around 200 microvolts. So this is how where you see is a K complex is that where it occurs spontaneously, but in order to a sudden stimulus when a person is sleeping or during sleep. Then you have lambda waves. So these lambda waves are actually monophasic waves. I'll show you what these lambda waves are, how they look. And these are positive waves, again the sharp waves, but these actually occur from your occipital lobe. So this we saw in the very first week that, okay, we have the central nervous system, and then we have the frontal lobe, temporal lobe, parietal lobe, we have the occipital lobe. So there were different lobes which are associated with different activities. But just to get a quick revision, that this is responsible for the visual cortex, because here only you have all of your visual connections that are connected through, and it goes to the optic nerves and fibers, everything. So allowing you not only to see but also to process the information from the external world, but also to assign some meaningful information and to make so to remember these visual perceptions or to make some interpretations from them also. So it's dedicated highly to vision, but some other complex functions are also there. So okay, it's obvious that if you see something, you actually know that, okay, what the size of it is. Okay, from size, I'll give you some what do they say, I forgot. It's like, I don't know, there was a name for it, a riddle kind of thing. It's not actually a riddle, but okay, a trick question, that if I have these one and if I have this, which one has a longer length? So this is actually given by someone called as Muller. This is when people perceive different things in different way. Or yeah, the action. So I don't know. It's visible, right? Yeah. So with 1 and 2, which one has the longest length? I know this is not related to your curriculum, but it will only help you remember certain things.
Ma'am, we can't answer it without any reference.
No. So just assume that the if I give you the scale, it would. It's just that you need to see and say. It's not like, okay, I'll give you a scale, and so it's not. If you in normal daily life, if you just see things around, or there's a pillar post standing, you won't go with the scale and see the size of the pillar post, right? You just know that, okay, that's taller than you. So that way, this is something for perception related studies.
First is longer.
Okay, so if you see first one, you do it like this, take the second one, do it like this. So actually the way you perceive things, so both are same. One is tilted outwards, one is tilted inwards. That's it. The length of them both is same. The length segments are same. So that's how our occipital lobe is involved in what it does. So this is something was given by Muller. No need to pay more attention to it. It was just something. So again, it helps us to map something, to assess the distance, size, and depth. Now, how depth is something that we have in third dimension. So normally what people, we or in 2D we live in, that is, but 3D objects are all around us. So we have this X entity, we have this Y entity, but we also have this depth or Z entity. So so we have something like this. This is X and Y. But we also have something like this, that is Z, that is depth. So we are able to visualize what's the depth of it. But somehow we have all of this in our cells. We have the eye cells. There are these rods and cones in the fovea, in the periphery of the retina. We are able to visualize these things very nicely. In order also to determine the color properties of them. Different functions, say for the object movement while we are reading, to differentiate from the information that, okay, what this particular information was all about, and then to identify a visual stimuli. Okay, if something's going on, you know that if it's not a ball that is hurting you, or if it's a stone hurting you, you'll know that what you want to do and what sort of action that you'll have to take for that. So that's what your occipital lobe does for you. Okay, so this is something called as mu rhythm over here. So I'll just show you what those lambda waves were here. So these lambda waves have something, if you see nicely over here, so their magnitude changes. So this wave is like this, whereas this wave over here where it went. Where it went. So this coming on this way. Something happened here. Okay. We should not zoom it too much. So this particular complex here and this particular complex here, if you see the difference in between both of them, you'll be able to see that, okay, I'll share the notes again, you can zoom it in and see that there's a phase reversal of the positive discharge. The actual wavelength, the signal, it changes its own orientation. So that is how you differentiate for waves that are called as lambda waves. And there are something called as mu waves, which is something like the symbol that you all know, that's a mu, 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 mu. So it occurs like this. Most of the waves that you see over here are like mu waves, but there are something differences over here. These are something called as sharps and spikes. So these are slow moving waves. So we'll see what these are. Okay, here it's only there. So spike and sharp wave. So a spike wave is something that a sudden rise. So if I ask you that, okay, how do you differentiate a QRS complex? Is it a spike or is it sharp? So given from the millisecond time duration here, it's 70 to 200 millisecond, and this is less than 70 millisecond. Is the QRX complex sharp or spike?
Spike.
Yeah, yes. So it's just that you get the actual direct information from it, and you see that it's like lesser time interval also. So these spike waveforms, they are actually used as a precursor for epileptic studies. So what happens is that when a person is suffering from epilepsy, I hope you all know that what epilepsy is, a condition where people can't remember certain things, or they do have some fits or attacks. So there are some seizures that are going through in their own body. This is something where your central nervous system is not performing well. There are some abnormalities going on there with a certain lobe, and that particular lobe can be, say, hypothalamus, or can be the thalamus region. Mostly these neurodegenerative disorders are associated with the hypothalamus region. So what happens is that the period where a person is suffering from that seizure or uncertain movement and activities, we see that that particular period is called as an ictal period, and the period just before that is ictal period is called as a pre-ictal period. Now this pre-ictal period, so if if someone is doing, so for example me, I did a project as to differentiate so like to find out the onset of this seizure. So how do I did that is like I took an hypothesis that if I am able to find out this beginning of this pre-ictal period when actually the seizure is not happening, but we are actually able to see some features of the seizures, there are some abnormalities in the signal, then we'll be able to differentiate the pre-ictal period and the ictal period. So that is how people do is like, and the seizures are, they are more specifically given by sudden spikes. So these spikes help us to characterize these features well. So then there's the sharp wave that's more longer than the spike wave, and it's also more similar. So here, if you saw, these were all sharp waves, whereas the signal before it, here there is one that's a spike wave. Guess there are not much of them. So again, here if you see, this is a spike wave, but can be a noise also. So again, there's a slow wave, so it goes on if there's a person is sleeping, and so this kind of wave will occur over there. So again, which type of waves are where? Okay, so something like this sharp waves, the sharp waves in the dorsal region, how they are, in the ventral region, how they are. So I suppose you know dorsal and ventral is like front and back. Then there are ripples again, how it varies in between the dorsal region and in the ventral region. It's just because of the way our body is, the way our systems are. So it's like this. Again, for the complex spike burst, it's something like this. So there are these different types of waves which are just given the name in a way that they look. That's it. Then there's this spike wave, and the it's like single spike and a single wave and multiple spikes in a wave. So sharp wave is like just a single wave, whereas spikes are these sparks that you get over here, a peaky. And spike in a wave is like you get a spike and then you have a wave. If you get a spike, then you have a wave. You just need to know that how they are. You don't need to know that, okay, how it varies or not, just a physical interpretation of it. I guess sir might have in the slides might have shown that, or might have stated the millivolts and the time seconds for each of them. Like here, these ones, you can just go through them, that would be nice one. And also the pathology they are associated, or the ones where they will be seen into. And poly spike and a wave, so multiple spikes and then a nice wave, so it's like this. So then we have read what EEG was, and now we'll see that okay, how do we do rhythm detection in EEG? Like how do we, it's like now in EEG we no longer have an event over here, we have a rhythm, because is everything very periodic. We have sudden waves, multiple waves, there are more samples we have over here, and everything in nerve conduction is very random. You won't actually differentiate, it's just the time seconds, the frequencies that you have in your hand, and you'll be able to differentiate using that. So there are two popular algorithms what people do over here. I guess we have seen them previously. First is the cross correlation. So we saw that what cross correlation was, that is the dot product of the two signals, since it's cross correlation, not autocorrelation. One at a particular time instant, other the delayed or advanced version of it. Okay. So here it's like just a simple basic equation of cross correlation, and how the cross correlation, this coefficient is being defined. That is x of n, y of n plus k. That is both the signals multiplied to each other and taking the average of them. And then the energies of both of them divided by energy, so that we normalize it. That's it. Square root of it. Then there is this another technique called as cross-spectral techniques. So as in cross correlation, before just looking into the time domain, here we look into the Fourier domain, that is the frequency domain. So we take the Fourier transform of the correlation function, whether it's an autocorrelation function or it's a cross correlation function. If it's a cross correlation function, it is called as this one, that is normalized coherent spectrum. But if it's, or it's also called as this cross power spectral density, there are some terms and like syntaxes for each of them, the semantics vary. So what does this power spectral density does? How we saw, I guess, in week 2 or week 3, that how do we find the power spectral density? We take the Fourier spectra and then we square the particular signal, so that we no longer have the magnitude variations, but we are able to differentiate in between the phase, able to exploit the phase. So it shows us the frequency at frequencies which are corresponding to the periodic activity. So you might want to notice this changes over here, because it will change in the certain ones, and it shows us the activity over a band. So you took a Fourier transform of the signal that's in time domain. So when you do actually, so you can actually write the Fourier transform of the signal with respect to the Fourier transform of its own conjugate. That's nothing but using property. You see that the magnitude of the entire own signal itself, square of the magnitude. So this is for the amplitude spectra. What you get is, next one you do is like you take the two other signals. So here was autocorrelation function of it. Now we do the cross correlation function of the same. Similarly, so what it does is like here, what we saw that it, since it was two same signals, what we saw that it will give us information into the periodic activity, corresponding to the periodic activity. I guess it's not visible there. To the periodic activity. But here, in this one, what it gives us, which are present in both signals that are being compared, the peaks in both of these signals, because now we are using two signals, so there will be two different properties. So it's similar to that, how we do correlation, that okay, say if you have a triangular wave and you have this kind of wave, if you do correlation of it, this triangular wave peak point, when it's moving through this, it would be maximum, but it's moving through this point, there is this delay edge here that's it going through, so hence the waveform would vary. So in it will give us the signal that's present in both the signals when it is compared. Then there's, so apart from correlation, we have this something called as normalized coherence spectra. Now what does this normalized coherence spectra does is the same equation for the same okay, sorry, this is S. So it's again given by the cross correlation function's magnitude divided by individual autocorrelation by square root of them. So how do we find the phase of it? That's taken the phase of this particular signal, you get the phase of this particular this normalized coherence spectra. Now this was two type of basis or methods that were based on correlation. The other one that we are going to see is also based on correlation or some slight modification of correlation. So new term is there that's called as match filtering. Now I'll tell you one what do you say, example for match filtering, so you'll be able to correlate it how this is varying. So you know there's something called as super resolution imaging. Has any one of you heard about that? Or say bubble imaging. Bubbles are something that we use as contrast agents. So what they do is, like, if you have say if I'm monitoring blood flow through a particular artery or vein, but there's very little contrast through it and I'm not able to see, so what we do is, like, we inject some bubbles. These are some chemical molecules which went into the body, and when we image them, they gave us a significant level of contrast. Now this contrast, we can, it's like we can, what do we say, we can image it properly in order to see where the blood flow is like, either it's towards the transducer, away from the transducer, if it's lesser or higher. So in a way it varies. So what super resolution is, breaking the limits of resolution. So when you actually break the limits of resolution, you're able to see beyond the, no, sorry, beyond the resolution limits of the system of the, through which you are imaging. So you're able to break the diffraction limit over there, where the wave bends and diffracts again. So what? Why do we use the match filter? Is that, say if you have, so I normally said that, okay, we use the contrast agents in order to see if we are not able to detect the blood flow through it, because the blood flow is very less or very small when we go through the micro vessels. So there, what we do is, like, we do match filtering. What we do is, like, take a particular region of interest, see how the bubbles are moving through that particular area. Now we want to correlate with some other area out there. We'll take the same region and we'll compare it with that, say A, this was region A and this was region B. We compare it with the region B and see how the region B is varying. So this is how we are matching the spectra of both of these regions. Now you can also say that this is something called as cross correlation, but I do argue that it's autocorrelation, because the same blood is flowing through, but the particles of blood are disoriented. So everything's not in proper orientation, hence the signal won't be coherent. So what you get is random activity. Hence you're able to see a correlation in between these random activities, and you're able to see where the magnitude is higher and lesser. So it's something like this. So it's nothing but matching two things here and there. So sorry to give you most of the jargon, but it's something that's matching things. So something that repeats, an event that repeats, has same characteristics. It can be used when a sample or template is there. So you know something is there, you have prior information of it, then only you can apply this kind of filter. You can't directly apply it on. And it's used to detect the event that's buried in noise. You can, it's like, if I have some bubbles which are, say, some different x diameter, but if I have bubbles, like, say there's a bubble which is of some chemical composition, but I do not have a bubble with a chemical composition, that's just an air bubble, it just made up when it was moving through our blood vessels. Because you might have noticed, when we are injected in something, they do like tick tick, it's like they try to remove the blood, it's like bubbles from it, so that it doesn't go into our body, and there's no thrombosis or blood clots or something like that does not happen inside. So for the same thing, what people do is, like, if so, now what they are trying to say here, that it can able to detect the event that's buried in noise. Now here the air bubble is a noise for us, but the bubble that's with the chemical, that's a nicer bubble for us. So it can actually see that, okay, which is more denser, which is more not denser, which has something properties there, or if it's a filled one. So there only we will be able to see, and if it's okay, this is, it doesn't have anything, it's empty or it's null, so it will directly differentiate it from the one that is we are trying to look into it. So it's basically a correlation filter in the end. So what do we do? So we all know, if we take a signal, say x of t, and an impulse, say h of t, we take the Fourier transform of both of them, we get x of f as well as h of f. If you want to find the y of f, that's output, that's h of f times x of f, where h of f is the transfer function. If we do y of f by x of f. So output energy can be maximized if we take h of f as k times, k is again a scale factor over here, x conjugate of f. So when we take the conjugate of the particular signal, so keep in mind, okay, this is the conjugate, not the signal itself, and exponential of negative j 2 pi f t naught. So here we have flipped the time axis. The time axis is no longer the positive time axis, we have actually flipped it, hence the delay, and we represent the delay by t naught over here. So this is how we say that, okay, the output energy would be maximized when the transfer function would be expressed in the order of something like this. So I guess that's it. Match filtering was the last topic and we're done. So do you guys have any doubt in EEG? Or event detection in EEG? If not, we'll go through the assignments, the 10 questions, that's it. No doubts? can give thumbs up if you don't want to say, if you do not have any doubts. Okay. Okay. Who else? Okay, then let's go to the assignments. Then that's a previous assignment that should help you do this assignment. So I guess most of you have done, so it's fine. I guess it's visible. Present on this device. Grant permission. Sorry. Okay. The screen is visible?
Yes,
Yes,
ma'am.
ma'am.
Okay, okay. So first question. QRS wave in ECG waveform is the result of okay. We'll go through the ones who have their mics muted for a long time now. Who can say? Srinaresh, can you? I know all of you here know the answers for these questions. You all have that level of knowledge, so it's fine. Even if it's wrong, it's not a issue for me because I'm also a student. I make mistakes, so just go on. QRS wave in ECG waveform, that's a result of relaxation, ventricular relaxation, or contraction of atria by AC node impulse, or simultaneous contraction of ventricular muscles by the stimulation of Purkinje fibers, or none of this.
Ventricular relaxation.
QRS. Think twice. Someone else want to try? So we saw P wave for was some depolarization. QRS was some depolarization. T was some repolarization. So what is depolarization? Depolarization is something when something contracts, and repolarization is when it relaxes. There are some other names for it also, that is systolic and diastolic. So this is for the answer is C. That's the simultaneous contraction of ventricular muscles by the stimulation of the Purkinje fiber. So the blood, it flows through and into the systemic circulation. Okay. Then next question. Contraction of atria by SA node impulse produces in an ECG signal. So when the atria depolarizes or contracts, which wave is being generated? Okay. Shravani, can you answer this?
I think it's a P wave.
Which?
P
P
wave.
wave.
P, yeah. Next, which of the following is important for recognizing the baseline of ECG signal? P wave, QRS wave, PQ segment, or T wave? We went through this. Come on, guys, unmute yourself. There are just 7 questions left.
C, PQ segment.
Yeah, I guess so. Yes. Then why do derivatives-based filters work best for detection of the QRS wave? That's a peak detection. Why do they work nicely? I guess all options are more or less good options. I'm also confused with the answers over here. What do you guys think?
It's the B option.
Which one option?
C, QRS complex has a
think
large
B.
slope.
I
Can
think
be.
B.
What were you saying, Ayush?
Ma'am, B option.
Works well for slow varying signals. Okay. Okay.
And
What do
because
you
in
think for D?
Penn-Tompkins algorithm, and because in Penn-Tompkins algorithm, the derivative part removes the P and T wave actually. So I think
It was not actually removing the P and T wave, but it was removing the low frequency components. So can also be the low pass filter option. Let's see which option. So answer is C. Okay. That's it has a larger slope. Oh nice. Okay. Who was like, I guess someone said the answer for that. Okay. Next, integration in Pan-Tompkins algorithm does what? So suppresses the low frequency. I don't think so. Makes the differentiators result positive. Merges multiple peaks into a single peak. Emphasizes differences resulting from QRS complexes.
Merges multiple peaks into a single peak. B.
Yeah. Yes. Then dichrotic notch can be detected by, oh, we didn't see that. The same algorithm. Okay. Searching for the highest peak obtained using the derivative filter. Searching for the highest peak obtained using the derivative filter and moving average filter. Searching for the
B
highest.
option.
Which one?
And D.
T. For the second peak obtained using derivative filter and moving average filter. What is the second peak here?
S2.
Oh, okay. I got confused for a time. S1 and S2. Okay. You all got why, what was the difference over here? Just remember that, okay, where the dichrotic notch came into role. So, okay, that is S1 and S2 and how it varies, and there only you'll get the difference. Okay, good one, Ayush. So, if power spectral density of an EEG wave is used to detect the presence of alpha rhythm, then is used to detect the, okay, which I guess it is asking something that where does the peaks for alpha rhythm occur, or what indicates, which particular frequency range indicates the presence of alpha rhythms.
8 to 13.
8 to 13. Okay. Yeah, yes. Okay. The minimum and maximum values of a correlation coefficient are. I guess we saw this was not there in today's.
1 and 1.
Sorry, what? Which one?
-1 and 1.
You know the answer for all of them, huh? Okay. Ninth question. I guess you'll have to solve this. A differential filter is given by y of n, that's x of n minus x of n minus 2 minus x of n minus 3, where both of them are input and output signals. We have x of n given as something. So y of n is equals to what? Try interpreting the signal. See.
A option.
Okay.
A option.
Okay. So just give the equation. Was this there in this year's assignment?
No, he just checked the options.
Okay. So you find it's like input the indices there and then see the equations and just it's like manual computation. That's it. Last question. So an event detection system consists of derivative filter that followed by a moving average filter. So the derivative filter is given to us like this, and the average filter is given to us by this, both input and output. You want to see the answer? Oh. So assume the signal value outside the interval is as 0. So we do not have anything outside this one, and assume that it's starting from 0 over here at n equals to 1. So the first sample in both input and output is at n equals to 0. That is this one. Oh shit. Okay, sorry. Can try solving it. Do you guys get how do we solve it? You can just try. Let me know. I'll
D.
show you the solution. Which one?
D. Option D.
Yeah, yes. Can you explain to all of us?
Sure.
Yeah,
Put
go
the
ahead.
values at the n equals to 1. When you put the values, then you will get the yn as equals to x of 0. Because x of -1 is not given, so we will take it as 0.
0. Hmm.
So we get the y of 0 as 1.
Hmm.
And then for finding the z of n, put n equals to 0. So you will get y of 0 by 2 and y of -1 by 2. Y of -1 is not given. So we will take it as 0. And y of 0 by 2, as we have find y of 0 is equals to 1. 1 by 2 is equals to 0.5. So A and D options are now the prominent options. B and C get rejected. Now let's check for the 1. Y of 1 is equals to x of 1 minus 2 of x0. Put the values. X of 1 is 2 and x of 0 is 1. That is y of 0 is equals to, y of 1 is equals to 0.
Okay.
Now find the z of 1. So y of 1 by 2 plus y of 0 by 2. And we have find that y of 1 is 0. So y of 0 by 2, that is 1 by 2 is equals to 0.5. D is the answer.
Good. Okay, that's it. We also did it like this. So, okay. If any of you have any questions, you can ask me. Thank you.
Thank you, ma'am.
Thank you. Okay. You have any doubts, Saish? Actually, you're not audible. You can try removing your headphones or so. It's coming like click, click, click. Are you trying to say something? You're not on.
No.
Huh?
Because I joined late, so could not cover the this video. Please, can you hear?
Sorry, my internet got disconnected. So I was saying that if you have any questions, you can ask me. Afterwards, like the recording, I've recorded the entire session, so it will be uploaded on YouTube, so you can see there.
Okay. Thank you.
Thank you.
Thank you.
Thank you. If you do not have any doubts, you can leave the session. Thanks. Thanks for
Can
joining.
you say the frequency range for different cases, ma'am, while at waking with a relaxed state?
Sorry. Your voice was getting cutted in between. Can you repeat again?
Can you say the frequency range for different cases while waking up at relaxed state with an activity like that?
Your voice is not that audible. Can you say it again? Sorry.
Now it's clear now.
It's cutting in between. Are you in a good internet zone?
Marina audible.
You can try saying it again.
Can you see the frequency range different cases, ma'am, like waking up at a relaxed state or an activity?
You want to see the frequency ranges
No, but relaxed state like that.
for EEG, right? These ones.
No, ma'am, general frequency range, ma'am, like
So normally when a person is relaxed, we say that it's a alpha waves that we are concerning into. So that's normally 7.5 hertz to 12 hertz. But again, it depends that what you want to see, that if it's a normal state or walking state, then it can be also beta waves. So depending on the activity that you're actually trying to see, it can vary.
Yeah.
Thank you.
Thank
Okay,
you.
do you have anything else?
No,
Okay, thanks.
thank you.
Mm.